Welcome to Biomed Explorer. In this video, I'd like to talk about something so small that roughly 10,000 of them could fit across a single grain of sand, and yet inside that impossibly tiny space is a fully functioning, self-sustaining city, the human cell. I'll cover what a cell actually is, what it's physically made of, the wall that decides what gets in and out, the control center running the whole operation, the machinery that builds proteins from scratch, the factories and shipping departments that finish and deliver them, the power plants keeping the lights on, the cleanup crew, the internal skeleton holding everything in place, how cells talk to each other, how a single cell becomes two, and what happens when any of this machinery starts to break down. So, let's get started. What is a cell, really? Most people know that cells are the building blocks of the body, but here's something that rarely gets explained properly. A single cell isn't a simple blob of goo with some stuff floating inside it. It's closer to a miniature self-operating factory, complete with a command office, a research library, production lines, packaging departments, power generators, a security wall, and a cleanup crew, all crammed into a space you'd need a microscope to even locate. The human body is made up of an estimated 30 trillion of these factories and roughly 200 different types, each specialized for a different job. A red blood cell is shaped like a flattened disc built to carry oxygen, and it's so specialized it actually ejects its own nucleus during development to make more room for that job. A neuron is stretched out like a thin cable, sometimes several feet long, built to carry electrical signals over long distances. A muscle cell is packed with fibers built purely for contraction. Different shapes, different jobs, but almost all of them share the same basic internal blueprint, and that blueprint is what we're going to take apart today. What a cell is actually made of. Before we tour the individual structures, it's worth pausing on what all of this is physically built from, because it's a surprisingly short list of ingredients doing a surprisingly large amount of work. Water makes up the majority of a cell's mass, acting as the medium every reaction takes place in. Proteins do most of the actual labor, folding into precise shapes that let them act as enzymes, structural supports, or transport channels. Lipids, or fats, form the membranes that wall off every compartment inside the cell. Nucleic acids, DNA and RNA, store and carry the instructions for building those proteins in the first place, and carbohydrates provide quick energy and, on the cell's outer surface, act as identification markers other cells can recognize. Four basic categories of molecules, arranged and rearranged in different combinations, are what separate a living, functioning cell from a random puddle of chemicals. Now let's see where each of those ingredients actually gets put to use. The plasma membrane, the wall that decides everything. Let's start at the outer edge, since nothing gets in or out of the cell without going through this layer first. The plasma membrane is often pictured as a simple bag holding everything together, but that undersells it considerably. It's built from a double layer of phospholipids, arranged so their water-loving heads face outward toward the watery environments on either side, while their water-repelling tails huddle together in the middle, forming a barrier that most water-based substances can't easily cross on their own. Scattered throughout that fatty layer are thousands of embedded proteins, some acting as gates that open only for specific molecules like sodium or glucose, some acting as receptors that detect chemical signals arriving from outside, and some acting as identification tags that let the immune system recognize the cell as belonging to the body rather than to an invader. This entire arrangement is often called the fluid mosaic model. Because the membrane isn't rigid, it flows and shifts like a thin layer of oil with proteins drifting through it, constantly adjusting to whatever the cell needs at that moment. Cholesterol molecules are tucked in between the phospholipids too, acting almost like a stiffening agent, keeping the membrane from becoming too rigid in the cold or too loose in the heat. It's less like a brick wall and more like a nightclub bouncer who's also somehow the building itself, deciding in real time who gets in, who gets out, and who never had a chance to begin with. The nucleus, the command center. Now let's move inside, toward the structure that runs the entire operation, the nucleus. If the cell is a factory, The nucleus is the head office, and it's usually the largest and most obvious structure inside, sitting near the center, wrapped in its own double membrane called the nuclear envelope. Inside that envelope is the cell's DNA, the complete instruction manual for building and operating a human being, coiled up into structures called chromosomes. The nucleus doesn't just store this information, it actively uses it, deciding which genes get switched on or off, and in doing so decides what kind of cell this will become and what job it will perform. A skin cell and a liver cell carry the exact same genetic instructions, they simply read different chapters of the same manual. A small, dense region inside the nucleus called the nucleolus works almost like its own department, dedicated entirely to assembling ribosomes, the machinery the rest of the cell will use to build proteins. Here's a detail that surprises a lot of people: the nuclear envelope isn't sealed shut. It's dotted with thousands of tiny pores, nuclear pores, that act like a security checkpoint, letting genetic instructions out and letting certain proteins in, while keeping the vast majority of the cell's contents strictly separated from its most valuable asset. Ribosomes, the protein builders. Once instructions leave the nucleus in the form of a molecule called messenger RNA, they need somewhere to actually be read and turned into a finished product, and that's the job of the ribosomes. These are among the smallest structures in the cell, and unlike most of what we've covered so far, they aren't wrapped in a membrane at all. They're simply dense clusters of protein and RNA. Ribosomes work like a construction crew reading a blueprint line by line, stringing together amino acids one at a time in exactly the order the genetic instructions specify, until a complete protein chain is formed. Some ribosomes float freely in the cytoplasm, building proteins the cell will use internally. Others dock onto the surface of the endoplasmic reticulum, which is exactly where we're headed next. The endoplasmic reticulum, the production and transport line. Surrounding the nucleus is a winding network of folded membranes called the endoplasmic reticulum, or ER, and this is where the real manufacturing begins. The ER actually comes in two distinct forms, doing two very different jobs. The rough ER gets its bumpy appearance from thousands of ribosomes stuttered across its surface, and this is where proteins destined to leave the cell, or to be embedded in a membrane, get built. As each protein is assembled, it's threaded directly into the ER, folded into its correct three-dimensional shape, and prepared for its next stop. The smooth ER, lacking those ribosomes, specializes instead in building lipids, processing fats, and in liver cells specifically, breaking down toxins and drugs before they can cause harm. It also plays a major role in muscle cells, storing and releasing the calcium that triggers contraction. Think of the rough ER as the assembly line building the product, and the smooth ER as the separate chemical processing plant next door, both feeding into the same shipping department. The Golgi apparatus, the packaging and shipping department. That shipping department is the Golgi apparatus, a stack of flattened membrane-bound sacs that sits close to the ER, ready to receive whatever gets sent its way. Proteins arriving from the ER get modified here, tagged with chemical markers, often small sugar molecules, that function almost like a shipping label, telling the cell exactly where each protein needs to go next. Once a protein is properly labeled and packaged, the Golgi apparatus buds off a small membrane-bound sac called a vesicle, loads the protein inside, and releases it to travel toward its destination, whether that's the plasma membrane, another organelle, or straight out of the cell entirely through a process called exocytosis. It's a remarkably precise operation for something happening inside a structure you'd need a powerful microscope to even see, and a single Golgi apparatus can be processing thousands of different proteins at once, each one correctly labeled and routed without ever getting mixed up. Mitochondria, the power plants. No part of the cell gets more attention outside of biology class than the mitochondria, and for good reason, since almost nothing else in this factory can run without the energy they produce. Mitochondria have a distinctive double membrane structure, with the inner membrane folded into ridges called cristae that dramatically increase its surface area. Along that folded inner membrane, the cell runs a process called cellular respiration, converting the chemical energy locked inside food into a molecule called ATP, the universal energy currency every process in the cell ultimately spends. Muscle cells and neurons, which burn through enormous amounts of energy, are often packed with thousands of mitochondria each, while cells with lower energy demands, like certain skin cells, get by with far fewer. Here's a detail that still surprises people who learned about mitochondria only briefly in school. They carry their own small ring of DNA, separate from the DNA in the nucleus, a leftover clue suggesting that mitochondria were once free-living bacteria that were absorbed by an ancestral cell roughly 2 billion years ago and simply never left, eventually settling into a permanent partnership that every one of your cells still depends on today. Peroxisomes, the specialized detox unit. Working alongside the mitochondria but often overlooked entirely are the peroxisomes, small membrane-bound sacs that specialize in a narrower set of chemical jobs. Peroxisomes break down fatty acids through a process that produces hydrogen peroxide as a byproduct, which sounds dangerous, except the peroxisome also contains an enzyme called catalase that immediately neutralizes it into harmless water and oxygen. In liver and kidney cells particularly, peroxisomes also help detoxify alcohol and other harmful compounds, working as a smaller, more specialized partner to the detox work already being done by the smooth ER. Lysosomes, the recycling and cleanup crew. Every well-run factory needs a cleanup crew, and inside the cell, that job belongs to the lysosomes, small membrane-bound sacs filled with powerful digestive enzymes kept at an acidity level that would damage the rest of the cell if it ever leaked out. Lysosomes break down worn-out organelles, misfolded proteins, and foreign material like bacteria in a process called autophagy, essentially the cell eating its own damaged parts to recycle the raw materials for reuse elsewhere. In white blood cells specifically, lysosomes take on a defensive role, engulfing invading bacteria through a process called phagocytosis and dissolving them before they can cause an infection. Without a functioning cleanup crew, damaged material simply piles up inside the cell indefinitely, which is exactly what happens in a group of rare but serious conditions known as lysosomal storage diseases. The cytoskeleton, the internal scaffolding. Everything we've covered so far needs to stay in roughly the right place, and that job belongs to the cytoskeleton, a network of protein fibers running throughout the cytoplasm that most people never picture as an actual physical structure. It's built from 3 types of filaments, each with its own role. Microfilaments, the thinnest of the 3, provide structural support and drive movement, including the pinching motion that splits one cell into 2 during division. Intermediate filaments act as tension-resistant cables, anchoring organelles in place and helping the cell withstand mechanical stress. Microtubules, the thickest and most rigid, function as internal railways, guiding vesicles and organelles to their destinations, while also forming the spindle that pulls chromosomes apart during cell division. Closely related to the cytoskeleton are the centrioles, a pair of small cylindrical structures made from microtubules sitting near the nucleus. Their main job is organizing that spindle apparatus whenever a cell prepares to divide, making sure each new cell receives a complete and accurate set of chromosomes. Unlike the scaffolding on a building, the cytoskeleton is constantly being built up and torn down, rearranging itself within minutes whenever the cell needs to change shape, move, or divide. The cytoplasm, the medium holding it all together. Filling the space between all of these structures is the cytoplasm, a thick jelly-like fluid made mostly of water, along with dissolved salts, proteins, and nutrients. It's easy to overlook, but the cytoplasm isn't just empty filler. It's the medium where countless chemical reactions actually take place, and it's dense enough that organelles don't simply drift around at random. They're held in relatively organized positions by that surrounding cytoskeleton. How cells talk to each other. A cell rarely operates in isolation, and much of what happens inside it is actually a response to signals arriving from outside. Receptor proteins embedded in the plasma membrane act like mailboxes, each one shaped to receive a specific chemical message, whether that's a hormone traveling through the bloodstream or a signal released by a neighboring cell. When a signaling molecule locks into its matching receptor, it triggers a chain reaction inside the cell, sometimes activating enzymes, sometimes switching genes on or off inside the nucleus, sometimes changing which proteins the ribosomes are actively building. This is how a single hormone released far away in the body can cause a precise, coordinated response in cells it never directly touches. The message gets relayed inward, department by department, until the right action gets taken. The cell life cycle from one cell to two cells don't last forever, and most spend their existence moving through a repeating cycle. During the majority of that cycle, called interphase, the cell grows, carries out its normal functions, and carefully copies its entire set of DNA in preparation for division. When it's time to divide, the process called mitosis begins. The copied chromosomes condense, the centrioles organize a spindle of microtubules, and that spindle pulls one complete set of chromosomes to each end of the cell. The cytoskeleton then pinches the cell down the middle, splitting it into two separate genetically identical daughter cells, each one a fully functioning factory in its own right. This is how the body replaces skin cells shed every day, heals a cut, or grows a child from a single fertilized cell into a person made of trillions. When the system breaks down, given how many separate departments have to work in constant coordination, it's no surprise that when something goes wrong at the cellular level, the consequences tend to ripple outward. When mitochondria fail to produce enough energy, the result is a group of conditions known as mitochondrial diseases, which tend to hit the most energy-hungry tissues hardest: the brain, the muscles, and the heart. When the machinery controlling cell division malfunctions, cells can begin dividing uncontrollably, ignoring the normal signals that tell them to stop, which is the underlying basis of cancer. When lysosomes lack a specific enzyme needed to break down a particular waste product, that material accumulates inside the cell indefinitely, gradually damaging tissue over time. And when receptor proteins on the membrane stop responding correctly to a signal, the entire communication chain behind them can fail, even if every other part of the cell is working perfectly. None of these are single, isolated failures, since a cell is a genuinely interconnected system. A problem in one department rarely stays contained to that department alone. The main jobs of the cell brought together. So now that we've walked through every major structure hiding inside this microscopic factory, let's bring it all together. Between the membrane, the nucleus, the ribosomes, the ER, the Golgi apparatus, the mitochondria, the peroxisomes, the lysosomes, and the cytoskeleton, a single human cell performs three broad categories of work every second of every day. The first job is protection and control, the membrane deciding what enters and exits, the nucleus deciding what the cell becomes and does. The second job is production and transport, the ribosomes and ER building proteins and lipids, the Golgi apparatus packaging and shipping them exactly where they need to go. The third job is maintenance and renewal, the mitochondria generating the fuel that powers everything, the lysosomes and peroxisomes clearing away waste, and the cytoskeleton holding the entire structure together while allowing it to move, communicate, and eventually divide. Wrapping it up, there you have it. The human cell, a structure smaller than the eye can see, and yet built with the same division of labor as any functioning city, a command center, a research library, a construction crew, a production line, a shipping department, power plants, a detox unit, a cleanup crew, and scaffolding holding it all in place, all coordinating with neighboring cells and eventually copying themselves entirely from scratch. 30 trillion of these tiny factories, each one running this same basic operation, quietly building and sustaining every part of who you are. The cell may be invisible to the naked eye, but every single one of them, working together in the trillions, is what makes the rest of your body possible. Thanks for watching, and Biomed Explorer uploads a new science video every day. Please subscribe and share.